Okay, greetings everyone. We are looking at the overall chemical reaction or chemical formula for cellular respiration, the breaking down of glucose in your cells to produce energy. You can see the glucose on the left, C6H12O6. That is the chemical formula for one molecule of glucose sugar. It's combining in cellular respiration, it's combining with 6 molecules of oxygen, which you have to breathe in, and that's going to produce on the right 6 molecules of water, which you're going to eliminate from your body somehow, 6 molecules of carbon dioxide, which you're going to breathe out, and a bunch of energy. That's what cellular respiration is. It's the breaking down of glucose in your cells. It's the oxidation of glucose, or the burning of glucose, to produce a bunch of energy. This is a stick model, like gummy bears and toothpicks, of a glucose molecule. So the little C's are carbon atoms. This O here is an oxygen atom clasp, I like to call it. The H's are little wimpy hydrogen atom band-aids that are stuck on all over the place. So this is just a representation of one molecule of that glucose sugar, and it's combining with 6 molecules of oxygen. See that O with a double bond to another O? That's O2. That's the oxygen that you're breathing in. And that's going to produce, again, 6 molecules of water, 6 molecules of carbon dioxide, and about 686 kcals per mole, or you can think of it as 4 kcals per gram of sugar if you were just to eat pure sugar, pure glucose, pure corn syrup. If you took a swig of corn syrup, you would be gaining about 4 calories or 4 kcals per gram. Photosynthesis, which we will cover in a subsequent chapter, is just the reverse. It's the exact reverse of this reaction. It's where a plant or a bunch of algae out in the ocean are building sugar molecules, are building glucose, which they will then connect together. You know, glucose is the building block of starch and cellulose and chitin and lignin and everything that the plant kingdom is made out of. So plants have to build their sugar molecules by using the reverse reaction, which we call photosynthesis. So plants are taking in CO2 from the air, okay, carbon dioxide from the air, or if it's a plant in the ocean, if it's a little algae floating around in the ocean, it's taking CO2 that's dissolved in the ocean water and using the carbon in that CO2. That's what the C is. It's a carbon, and building these big sugar molecules and then combining them into the starch and cellulose and so on. Cellular respiration, we usually break into 3 steps. So you can start, you can think of you're eating food, you're chewing it, and your enzymes in your saliva are starting to break it down, and then it goes through your digestion. The carbs are broken down into individual glucose molecules, pretty much, enter your bloodstream, go to your liver. Your liver decides what to do with them. Your liver is a chemical factory, literally. But it decides whether to store the glucose or pass it on to the cells. And so once that glucose, enters your bloodstream, and a cell, let's say a muscle cell, takes in that molecule of glucose, it undergoes 3 steps, which we lump together and call cellular respiration. So glycolysis, that's kind of a fuel preparation step. It's breaking the glucose in half, basically. See, we depict glucose as C6, meaning it's a molecule with 6 carbons. it's going to break it in half basically into 2 pyruvates having 3 carbons each, and this is going to produce a small amount of energy. Then this pyruvate enters the mitochondria, the power plant of the cell. So this is still in this muscle cell. It's undergone glycolysis, which literally glyco means glucose, and lysis means breaking in half. It's literally busting the glucose six-sided ring-shaped molecule in half and releasing a net 2 ATPs, which I'll get to in a second. It's charging up a couple ATP batteries, basically. Then that pyruvate, or those 2 pyruvate, enter the mitochondria. And there they undergo what we call the Krebs cycle. And I'm calling this the furnace because the pyruvate is oxidized all the way down to CO2 molecules and it's producing lots of energy, loading the energy onto onto little transport trucks and sending those off to the electron transport chain, which is really a power grid, which has a mechanical turbine, and it's recharging the ATP battery. Lots and lots of them. This is producing like another 36 ATPs. So starting with step 1, stage 1, this is kind of like preparing the fuel for the furnace. So this is going on in the cytoplasm of this muscle cell. It takes in glucose and splits it into 2 pyruvates. That's all it's doing. It goes through kind of an investment phase where it takes 2 ATP batteries to get going, and then 4 ATPs are harvested, so there is a net gain of 2 ATP, so-called batteries. In addition, 2 of these little energy trucks, NADH trucks, are also loaded, okay? And so this is the chemical reaction. It's about 10 steps, okay? These are molecules. This is, you can think of as little toothpicks and gummy bears again. So starting with glucose, which is that 6-sided ring sugar, it takes, the investment of 1 ATP battery to get started, and then it works its way down the line. These are all sugars. See, glucose, blah, blah, fructose, blah, blah, fructose again. It works its way down and turns the corner. This is just a schematic representation. It doesn't turn a corner in your cell, okay? These are just stick figures on a page. They're just depicting how all these series of molecules are formed. And then right at the end, of course, down here at the lower left, is this molecule of pyruvate. You see it only has 3 carbons, 3 little letter C's on there. That's what the end result of glycolysis is. Plus, as I said, plus a net gain of 2 ATP batteries and some little energy trucks. So this is going on not in the mitochondria. We haven't gotten to the mitochondria yet. This is going on right in the cytoplasm of the cell. Okay, so now if an organism has mitochondria, and if it has oxygen, a supply of oxygen, some organisms are anaerobic. They don't use oxygen, okay? So if it's an animal, if it's a eukaryotic cell, then, for example, then it has oxygen. It has mitochondria, so it continues on into stage 2, the Krebs cycle. So in the Krebs cycle, this is in the mitochondria, this pyruvate, these 2 pyruvate molecules that you've just formed now continue getting oxidized all the way down to CO2. Here's the dude's name, Hans Krebs. He won a Nobel Prize for discovering all this. And so this is sometimes called the furnace. This is producing a lot of ATP. These little black circles represent carbon atoms. So when we draw a symbol like this, it's a schematic representation. And, you know, we're leaving off all the little hydrogens and incidental little oxygen sticking off. We're just showing the carbons. That's all we care about in a diagram like this. So in the mitochondria, the pyruvate enters as this molecule called acetyl coenzyme A, acetyl CoA, and then starts this cycle. It's not a circle. It's not going around in circles. Things aren't spinning. Your mitochondria aren't spinning. This is a schematic representation. We show it as a circle because it is a cycle of sorts. And this just shows how it gets broken down and produces even more ATP. This molecule, acetyl CoA, is formed from that pyruvic acid. Now, this is another way, yet another way that biochemists depict molecules. So we're showing bonds here. So look at that pyruvic acid. We're showing CH3 bonded to a central carbon, and that carbon has a double bond going up to an oxygen. So don't get confused. These are just different ways that biochemists use to depict, you know, these molecular structures. The O's are oxygen atoms. The H's are obviously hydrogen atoms. And this is forming this acetyl CoA. So this is really large and negative due to all of its phosphate. I'm reading down here. PO4 is phosphate. It keeps the acetyl CoA from going back out of the membrane and escaping from the mitochondria. It grabs onto it, hangs onto it. See this coenzyme A. If you want to know the why behind all this, it's a big molecule that grabs a hold of that and keeps it in the mitochondria, keeps it from escaping back out. It's a trick, or it's a carefully designed system. So we call ATP the energy molecule of the cell. So people use this money analogy or currency analogy. If one molecule of glucose was a $10 bill, ATP would be 25 cents. So ATP is a more usable size or packet of energy. You can't go to a video arcade with a $10 bill. You need quarters. So the same thing applies here. The ATP is sized just perfectly to power all the machinery in the cell, all these little machines and tools and mechanisms and walking motor proteins and all that. The ATP is just the perfect size to power all that. Glucose would be too big. It's like a $10 bill. Let's see, there's also a battery analogy. So the ATP phosphate bond stores, well, that many kilojoules per mole. We haven't covered that in class yet, but about 6 kcals per mole. These other energy molecules, the NADH, stores a lot more, like 7 times as much. Glucose stores way more, 2,800 kilojoules per mole, or about 686 kcals per mole. So way, way, way more. This is what ATP actually looks like. So this is yet another way of depicting a big molecule. You have a sugar, and it's connected to, it's bonded to the nitrogen-containing base. And then you have 3 phosphate groups. Phosphate is PO4. P is phosphorus, the element phosphorus. O is the element oxygen. That little double line is a double bond. All the single lines are single bonds. It's just a way of depicting this molecule. So the high energy bond is the one shown in pink here. It's the one that's breaking and reforming and breaking and reforming. And every time it breaks, it's providing, 30 kilojoules per mole, or about 7 kcals per mole. And every time it's rejoined, it takes that much energy to rejoin it. So you have ATP, and it gets used. It's like an ATP battery. And when it's discharged, when the battery is, discharged, it's known as ADP, adenosine diphosphate. It's the uncharged version of the battery. So then you stick on another phosphate and it's charged back up to ATP. So this goes on and on and on. In the little rotary turbine that's churning away at the end of the electron transport chain. So ATP and ADP are constantly cycling back and forth. Now this is a bigger molecule, the NADH. The stick figure is on the left. So this is, gummy bears and toothpicks. And the one on the right identifies what each of those atoms is. So NADPH has this phosphate stuck on the bottom, whereas NADH just has an OH group stuck on the bottom. So we're talking about little minute differences. Let's see. The NADH is the charged form of this molecule. Stores about 200 kilojoules. So this is like equivalent to 7 ATP batteries, roughly. And this is, when I say charged, this is all it is. It's just an additional bond. Look at the red arrow. The two molecules are all identical except for just an additional hydrogen stuck on the top of that little ring at the top. And that provides it with 200 kilojoules per mole of energy. And so NAD plus is the depleted battery form. Okay, as I mentioned, this is in step 2, the Krebs cycle, step 2 of glycolysis, of cellular respiration. It's taking place in the mitochondria. Now, stage 3, this is a really interesting process. It's called the electron transport chain. And again, this is still in the mitochondria. And this is where these electron trucks, these energy trucks, NADH and FADH2, are unloaded. They offload their electrons to what is, in essence, a power grid. Yes, it's pretty amazing. It's made up of individual protein assemblages that are moving around in the membrane of the mitochondria, passing these electrons down a chain, just like a power grid. And at the end, driving this turbine, which I will show you in a second. Here's the overall energy balance. You need to know a couple things here. Glucose enters at the top. So step 1, glycolysis. You need an input of 2 ATP batteries to get that started, but you're producing 2 net fully charged ATPs. Then it enters the mitochondria. Krebs cycle is producing 2 more ATP. Then it gets to the electron transport chain and what we call oxidative phosphorylation. That's spinning turbine. And it's producing tons of money. Tons, like somewhere between 36 and 38 ATP. So if you see a question on homework or a test, the overall production is somewhere between 36 and 38. This diagram gives it as 36. That's fine. And that's the production. Okay, so now we're going to look at some pictures. Why do cells need energy? Because they are factories, they are cities. They have all kinds of machinery, mechanisms They're a production plant. They're a factory. They have, these little motor proteins, kinesins, walking around, towing these big cargo vesicles full of newly manufactured protein parts. They are literally an automobile factory. That's the turbine spinning over on the right. That's the ATP leaving, the ADP coming in, and the ATP leaving. So let's take a look at some movies. Okay, so let me turn the sound off. This is looking at the edge of the mitochondrial membrane, and mounted in this membrane are all these, jillions of these spinning turbines. And they are manufactured from, I forget, 20 or 30 protein parts. They are manufactured, assembled in this mitochondrial membrane. They have, mounting brackets. They have a drive shaft. They have a turbine, which is powered by protons. They have a camshaft. Yes, if you've ever worked on an engine, you know what a camshaft is. These have a camshaft. It's not a bump. It's a camshaft. Let's use the correct terms. It's mounted in this thing, spins, 100,000 RPM, maybe not that fast, and cranks out ATP batteries. So each of the mitochondria in your, let's say, muscle cells are utilizing these mechanical turbines that are on, really small on a molecular scale. That's depicting the protons coming through and getting the turbine to spin. And then that's literally a camshaft. So this is made out of protein molecules, and ADP, a discharged or depleted battery, comes in red there, bonds perfectly to one of those subunits right there. A phosphate is attached to it, and it leaves as ATP. This is showing us glycolysis.
Let's take a closer look.
Let me put that on mute. So this is glycolysis. We looked at this in class. This is one molecule of glucose sugar. You see the 6 carbons? You can identify it as glucose by looking at the 6, by noticing it has 6 carbons. It's going to come in the cell, and it's going to be broken in half here. We have taken a look, or we'll take a look at this in class. That's water. It's going to get split in half. And there's the investment phase. So those 2 ATP batteries, I think is what that was trying to depict, are coming in. And voila, you have 2 3-carbon molecules, and it's going to go to work on those. That's an electron transport truck loaded up there with 2 electrons. Rather than sit here and go through that, we'll look at that in class. Now,
a large protein.
This is another look at the spinning turbine. And okay, so this is a closer look, and you can tell it's actually an assemblage of protein molecules, and it's spinning. This is greatly slowed down. The yellow little thingies are protons that are going through it and causing it to spin, like, I want to say 7,000 RPM or something like that. And you can see the ADP, the depleted batteries, entering, and the ATP, the charged batteries, leaving. See, the protons are doing the driving. They're the water flowing through the dam. The protons are doing the spinning. Now, let's see. We've got an even closer view here, I think, of the ATP synthase turbine. So yes, we have come up with names for each one of these protein subunits.
The F1 complex is outside of the membrane, while the F0 complex is embedded in the membrane. There's the stalk, including the delta and the B subunits.
So you can tell by the way this guy is talking we have, discovered a lot about these little ATP synthase enzymes, I guess you could call it. I call it a turbine. And we know exactly how it works.
The subunits are seen as a ribbon diagram.
So a ribbon diagram is just a way that biochemists depict these big, massive protein molecules.
If we zoom into the active site, we can see the molecular structure of the...
Okay, so that's a depleted battery, ADP. It just bonded in a little pocket right there in that particular protein. You can tell it's ADP and not ATP because it only has two phosphate groups over on the left. These thingies, that's one phosphate, that's another phosphate. So you're about to see the third phosphate get added to it.
Here is ADP with its two phosphates. They are all held in place by a network of hydrogen bonds mediated by the amino acids in the folded structure of the beta subunit.
Okay, so this is why we spent so much time going over bonding and hydrogen bonding and everything, because that's how the world works. That's how cells work, through electrostatic interaction between molecules. It's very mechanical, and that's what makes it cool. So you have electrostatic attraction and repulsion going on between complicated molecules, and that's what makes life happen.
Base and the sugar, but really primarily the base. There's also a magnesium ion that plays a critical role in not just stabilizing, but also neutralizing the charge of the phosphates. Notice the conformational changes that are happening when the inorganic phosphate comes in. There is the promotion of a planar transition state.
Okay, so this is the third phosphate group coming in, getting ready to bond to that depleted battery and make it a charged up battery, an ATP, adenosine triphosphate. We know exactly how it happens, which is a testament to countless hours of researchers, because you can't see this through a microscope. These are little tiny molecules.
Synthesis of a new phosphodiester bond is catalyzed, and then the subunit switches to the open conformation.
So this is how an enzyme works. It bends a molecule slightly to get it to bond to something. It lowers the activation energy hump, if you want to think of it that way. It lowers the hill so things can bond together and sometimes break apart easier with less input of energy.
Upon this reconfiguration in the beta subunit, ATP is ejected from the active site. The enzyme then
Okay, so this is just an example, a great example actually, of how an enzyme works. In this case, kind of a complicated one, but it's very mechanical. It's taking that ADP, it's bending it a certain way, getting the phosphate to bond to it, and then releasing it. So now, with that information, we are ready to take a look at some of these homework questions. The first, 6 questions here are really easy. You can look it up in your book. But starting about question 7, 8, 9, we're talking about ATP. See, question 8, what is adenosine triphosphate known as? I don't know. There's like 5 different ways you can answer that question. You could say ATP, and that would be correct. You could say the energy currency of the cell. That would be correct. I don't know, you could say the battery of the cell. You would be correct. Okay, so there's a number of ways you could answer a question like question 8. The energy currency in cells, question 7. Okay, so we understand that now. Now look at 9 and 10. This is the reaction. This is showing ATP, the charged form of this energy molecule, being depleted. It's going to ADP plus phosphate plus energy. So that's a battery getting spent. Question 10 is a battery getting charged, a discharged battery, a depleted battery, an ADP, getting charged back up in that turbine. It's getting joined to one phosphate, which We just call P here. Yes, I know phosphate is PO4, but we're shortening it here. We're just calling it P. Plus energy is ATP, adenosine triphosphate. And muscle cell. Why do muscles need ATP? Well, because muscles need energy. Why can't ATP be brought to the muscles through the blood? I don't know. Probably wouldn't survive the blood. It doesn't work that way. Why? Because where would it be charged up? It has to be charged up in a rotating turbine. So there's a bunch of ways you could answer B there. Why can't ATP just come to the muscles through the blood? I don't know. There's 10 different ways you could answer that question. It has to be charged in a rotating turbine. We just spent 10 minutes looking at it. Those are mounted in the mitochondria. The mitochondria are inside the cell. They're not in the blood. They're in the cells. So bunch of ways you could answer that. Also, I think your book mentions that ATP is a fragile molecule and, wouldn't survive the blood or whatever. Okay, so a normal adult male. Okay, you can look this up in your book. Question 12. The point of question 12 is that you use a lot of ATP every day, more than you weigh, which is kind of crazy. And all that's pointing out is that ATP is constantly being regenerated, constantly. Millions and bajillions of molecules of ATP are being regenerated every second. Now, question 13. Why can't your cells just take their energy directly from the starch and lipids and carbs that you eat? That answer is right in your book, but I want you to think about it. We talked about it just a few minutes ago. The carbs are made of glucose. Remember, the glucose was like a $10 bill. It's too big. It's too big to power anything in the cell. You need a smaller unit of energy, and the ATP provides that. 14, I'll let you think about. 15, and basically all these are coming right out of your book anyway. I don't want to just give you all the answers. Okay, I will let you look up 15, 16, and 17 in your book if you don't know the answer. 18 is getting at the overall reaction for cellular respiration, which we talked at length about, and I gave you the answer to in the earlier lecture. 19 and 20 are some pretty easy true-false questions. Cellular respiration, it occurs in organisms that have cells. Okay, that includes pretty much all organisms. So it wouldn't take place in a virus. Viruses don't have cells. They're not made of cells. But it would take place in everything else. And so I'll let you look these up and think about them. Step 1 is known as glycolysis. We spent a bunch of time on this in the lecture. So just looking at these, these are pretty straightforward questions. C, D, the net energy production of glycolysis is. That's why I spent a bunch of time on that in the lecture. So go back if you missed that. 22, step 2 is known as the Krebs Cycle. Basic questions. Go back and listen to the lecture and read your book. Question 23. Step 3 is known as electron transport chain. Sometimes you'll see the term oxidative phosphorylation being applied to the whole thing. I like to use electron transport chain. And these are easy peasy questions we've covered. Okay, here's more of a case study question. Some biology students placed algae cells, which you can think of as an aquatic plant, in a covered test tube containing water and nutrients. If we haven't done this lab, we will do it soon. The beginning pH of the water was carefully measured at a certain pH, 8.0. We have talked about pH endlessly, and we have done at least one lab at this point on pH and on CO2 and how it affects pH. So think back to the lab. They placed the test tube in a dark box to prevent photosynthesis from occurring. And then they carefully measured the pH and plotted the results. So A, assuming that cellular respiration takes place in the algae cells, yes, it will. It will if they're alive, it will. Will the CO2 concentration increase or decrease? Well, to answer that question, look at the reaction for cellular respiration. We've only looked at it about 10 times in the last half hour. So go back and look at it. Is CO2 being produced, and would that end up in the surrounding water? Just think about it and use common sense. Now, part B, the students recalled from a lab. Yes, if we haven't done that lab, let's see, let me read this. Oh yes, we've done this lab a long time ago. Increasing CO2 levels in water lead to more acidity because you're forming carbonic acid, a dilute acid in the water. So it's going to get more acid slightly. So it's just asking a question and asking you to apply some logic, knowing a little bit about the pH scale and knowing that increasing CO2 levels in water lead to more acidity. So I'm not going to give you the answer to that, but it's pretty straightforward and logical. Now, these final questions are going to require you to watch a short video that's posted on the student portal. The time, I mean, I can't help you anymore here. I'm probably giving you too much help here. I'm telling you exactly where in the video to find the answers. So you should be able to do these last, 25 questions pretty easily by watching the video. Okay, that's it for this lecture. Let me know if you have any questions.